I'm so thrilled to join this new community of scholars and continue to put new science in the world, all for the goal of helping people all around the world. So thank you again to the ARCS Foundation and to the entire Washington Research Foundation for this fellowship. I look forward to seeing you all in the future.
Oh, by the way, this is my son, Henry. He's 3 months old. He's the main reason that we're so grateful for this additional support at this time, because daycare is really expensive, it turns out.
It's a real nice sense of validation to know that there are people that understand that the journey ahead is an arduous one, and that they're willing to provide financial support.
Your generous gift is so well received, and I truly appreciate it. It's going to make the next couple of years a little bit easier so that I can focus on my studies, so that I can hopefully become a pretty good plant breeder. And then after graduating, I would like to either work in academia as a plant breeder or perhaps in the private industry.
Welcome to the 2021 ARCS Foundation Seattle Chapter Luncheon. I'm Brittany Williams, a third-year ARCS scholar from the University of Washington, and I'm happy to welcome you to the annual ARCS Luncheon from Seattle Children's Research Institute, where I'm working in the lab of Dr. Ria Kohler on SARS-CoV-2 and Mycobacterium tuberculosis co-infection models. Thank you for taking the time to be here today to listen and engage with ARCS Foundation, which has been and will continue to be so valuable to me in my academic career. Before we go further, we'd like to acknowledge our generous sponsors, without which this event would not be possible. For those who may be new, ARCS Foundation is a nationally recognized nonprofit organization started and run entirely by women. Each chapter supports its local academic centers of excellence by providing unrestricted funds to advance cutting-edge research and science. ARCS chapters support students in science, engineering, and math, including multidisciplinary and MD PhD programs. Your support today will benefit future STEM scholars like myself along their scientific journeys. I am one of 168 fellows supported this year by ARCS Seattle chapter at the University of Washington and Washington State University. ARCS fellowships give universities the ability to recruit and retain thousands of STEM graduate students across the country. ARCS fellowships give students like me flexible funding to live, learn, and conduct research in Washington State. The generous members of the Fellows of the Seattle chapter have given over $21 million during the past 40 years. Nationally, over 62 years, ARCS has supported 11,000 students like myself through gifts of over $125 billion. Receiving an ARCS fellowship definitely influenced my choice to attend the University of Washington. I also had offers from UC Berkeley, Princeton, UT Austin, and Emory. I was leaning towards Seattle because of its large infectious disease research community and the possibility of networking with leading scientists in the field of infectious disease, but was concerned that Seattle is an expensive city to live in, and the unrestricted ARCS fellowship certainly took away some of my worries. It gave me the assurance I could come to Seattle and not have to eat ramen every day. For me and thousands of scholars, ARCS has given support that goes far beyond financial. ARCS Foundation led me to my mentor, ARCS member and leading vaccine scientist, Dr. Ria Kohler. I met Dr. Kohler at my very first ARCS event, and when I mentioned my interest in vaccine research, this sparked a conversation. Now I'm working in her lab. She provides me with ongoing opportunities to discuss and develop my practices as a scientist. Dr. Kohler has introduced me to industry leaders in my field. Being at the center of renowned research institutions like Seattle Children's, Fred Hutch, and the Bill and Melinda Gates Foundation during this pandemic has catapulted my research. Never imagined I would perform research on a disease such as COVID-19 while it was and is developing. I've been given the opportunity to be involved in processing samples associated with the Moderna COVID-19 vaccine to enable its rapid authorization and approval by the FDA, which has saved countless lives. On the preclinical side, I've been able to carve out my own project focused on two major diseases, COVID-19 and tuberculosis. I never would have had these opportunities had I not come to the University of Washington and joined this lab. Our renowned keynote speaker, Dr. Bedford, is a global leader in the pandemic and someone that I personally learned from as a member of the STEM ecosystem of universities, industries, and fellows. ARCS has also welcomed me into a community of scholars and scientists who share my passion. Becoming a part of this community gave me somewhere to start and somewhere to grow. Receiving this prestigious fellowship gave me a needed boost of confidence, combating what so many grad students face, imposter syndrome. As a first-generation college student, I never thought I would go to graduate school. By offering me a fellowship, ARCS Foundation saw the potential in me. It reminded me to believe I do have the skills and ability to succeed here. I'm very grateful to the ARCS Foundation for their many types of support. Later in our program, you will meet 4 additional ARCS fellows, Maya Lewinson, Avi Kenny, Shannon Allen Wiles, and Elizabeth Borgeson. They each will tell you their own story of how the support of the ARCS Foundation has positively influenced their careers. They'll also talk about how they are each utilizing the tools Dr. Bedford has developed in their own research to delve into and develop responses to pathogens. We are all very grateful to you for supporting current and future scientists through your participation today. It is now my pleasure to introduce ARCS Seattle Chapter President, Margaret Nyber McGann, who will welcome our keynote speaker, Dr. Trevor Bedford.
I am honored to introduce our keynote speaker for today, Dr. Trevor Bedford. Dr. Bedford is coming to us from the Fred Hutchinson Research Center campus, where he is an associate professor in the Vaccine and Infectious Disease Division, the Public Health Science Division's Computational Biology program, and the Human Biology Division. He's also an affiliate associate professor in the Department of Genome Sciences and the Department of Epidemiology at the University of Washington. Dr. Bedford uses powerful computers and complex statistical methods to study the rapid spread and evolution of viruses, including those that cause COVID-19, influenza, Ebola, and Zika. He co-developed an open source platform called NextStrain that provides a continually updated view of publicly available viral genomic data alongside powerful analytical and visualization tools. He is a leading advocate for the immediate release of research analyzing viral evolution during epidemics. Fresh information that can make a life-saving difference. Welcome, Dr. Bedford. We are so glad you could join us today.
Thank you, Margaret. It's my pleasure to be here today. So today I'm going to be talking about real-time tracking of pathogen evolution. And so how this works is we can now sequence viruses or sequence pathogens, look at the letters in their genome, and understand something about how an epidemic is happening. So basically, we can have this toy example where there's some individuals here, they're sick with, say, COVID, they're sick for, say, a week, and they cause a couple secondary infections, which cause their own secondary infections. But we can take some of these individuals, swab their noses, and sequence the virus out of these samples. And what we'll discover when we do that is there'll be patterns of shared mutations. So these 2 infections here share this purple mutation, these 4 infections share this orange mutation. And we can then take those mutations that we observe and reconstruct an evolutionary tree or a family tree or technically a phylogeny that shows how these infections are related to one another. And this tells us again something about the epidemic. So this has been useful in a variety of circumstances. Just as one example, we have the Zika epidemic in the Americas. This was published a few years ago, where we, with colleagues in Brazil and the United Kingdom, were able to sequence viruses from all over the Americas during the Zika epidemic, as well as the South Pacific, and estimate when Zika arrived and where it arrived to. And we find a single introduction from the South Pacific into northeastern Brazil at the beginning of January 2014. And what's remarkable here is this was a full year and a half before the first detected Zika case existed. So there's just all this cryptic transmission that was going on that we didn't notice, that was only observable by this pathogen genome sequencing. In another example, we have the Ebola epidemic in West Africa. So this was the largest Ebola epidemic up to this point. And we're able to, with these 1,600 pathogen genomes, virus genomes, understand about how it moved across this landscape. And so we see these kind of short-lived migratory transmission chains where it jumps from one village to another and doesn't persist in any place for very, very long. So generally this field has been making huge progress, but there historically wasn't really actionable inferences. So this would be published in a paper a year after the epidemic was over, rather than being really important while the epidemic was ongoing. So we sought to address this with this NextStrain project. This is all work in direct collaboration with Richard Nair at the University of Basel in Switzerland. And here's this project to conduct real-time genomic epidemiology and evolution analysis of emerging epidemics. And most recently, before the pandemic, we had operationalized this with colleagues at the INRB in the Democratic Republic of the Congo in Kinshasa, where they were able to each week, as Ebola genomes came off the sequencing machine, able to look at how they're related, look at transmission chains, and try to understand what's happening with the Ebola outbreak using this phylogenetic approach. And so, of course, the COVID-19 pandemic changes everything. But in terms of genomic epidemiology, there were really maybe 3 key roles and 3 key insights that the genomics have played. So one is this rapid human-human spread early on in Wuhan. 2 is extensive local transmission when testing is rare. And 3 is identification of variants of concern and tracking their spread. So if we go way back to January 11th, 2020, we have the first 5 genomes from Wuhan that are shared and sequenced by China CDC and others. These show a novel SARS-like coronavirus. We know that SARS is transmissible and highly pathogenic, so this is already worrying. But then I didn't really freak out until perhaps January 19th, when we had the first 12 genomes from Wuhan and Bangkok. These 2 in red here are the ones from Bangkok. And really importantly, we see that basically these initial genomes are almost identical to one another. There's just a handful of mutations that they have between themselves. And this lets us know that the original beginning of the outbreak, the beginning of the epidemic, started in November 2019. And by looking at this along with international cases, we're able to see just how quickly it had spread from that November 2019 beginning. And let us know there was rapid human-to-human transmission at a time when China was not even acknowledging any human-to-human transmission. So with this, share this with public health colleagues in the US and throughout the world, try to urge action, and try to provide that insight. So after the epidemic in China kind of starts to wind down in the course of January, February, during that time, we caught these international arrivals from China to the US. Here is each infection here in red. Those were caught and contained, but during this February time period, there are other arrival events that were pre-symptomatic or asymptomatic infections that were not caught at the borders. These caused local transmission chains to start and grow up into the US and European epidemics in March. So if we go back to March 2020, we were conducting the Seattle flu study at the time, swabbing people's noses who had a symptomatic respiratory infection and checking what virus was there and sequencing it. We were able to detect COVID-19 at the end of February, this first kind of community transmission before anyone else had noticed it. And by sequencing the virus, we're able to understand when it had arrived into Seattle, have that date be around February 1st, and really see how quickly it had been doubling, how quickly it had been expanding over the course of that month. So sharing this news results in kind of major mitigation response and has a really large effect on kind of the unfolding of the epidemic within Seattle and within the US. So march farther forward. At this point in the pandemic, really all of the focus is on alpha, beta, gamma, delta, et cetera, these variants of concern, and really all of this evolution that's been happening in SARS-CoV-2. And so here, now there's a massive global surveillance effort to look for these variant viruses and track their spread. At this point, we can see the emergence of these other variants, alpha, beta, et cetera, but at this point, everything is delta. There's some holdouts in South America, as delta is kind of barely starting to get there now, but the expectation is everything will be delta from this point forward. I'm now watching for new sublineages of delta to emerge. And with that, I'll end the talk. Really like to thank data producers from around the world, GISAID, the NextStrain team, and the lab. And with that, we'll continue the presentation. We have 4 ARCS scholars joining us today who are conducting research using the tools I've discussed. You'll now have the opportunity to go behind the scenes with each scholar to learn more about the research and how ARCS fellowships have impacted them.
My name is Maya. I'm a student at the University of Washington Medical Scientist Training Program, and also I'm a PhD student at the Genome Sciences Department. I'm originally from Portland, and I got a BS at University of Chicago in biology. And I really entered biology through a diversity of experiences that were really driven by my interest in understanding human disease. So I was originally really inspired by hearing a lecture from about Janet Rowley and her characterization of the Philadelphia chromosome. This is actually a abnormality that's found in a lot of leukemias, and because of that lecture, I actually applied to a competitive internship at Memorial Sloan Kettering Cancer Research Center. And there I really had the opportunity to work with Dr. Abdul Wahab to work on drug resistance in hairy cell leukemia. And because of that experience, I also wanted to continue in cancer genetics, and I worked with Dr. Lucy Godley at University of Chicago to further study inherited malignancies in leukemia. During my undergrad training, I also wanted to learn about diversity of approaches to study human disease and biology. So for example, I took an ecology and evolution course, and specifically I was really interested to learn about evolutionary approaches to study cancer genetics. And this really drove my interest in my PhD to study cancer evolution. So I was offered spots at several MD PhD programs in the country, including at University of Chicago and at Oregon Health and Science University. And although I didn't actually yet know that I'd become an ARCS fellow, it was really encouraging to know that there were opportunities to supplement the cost of living in an expensive city like Seattle. And when I did receive an ARCS fellowship, I was just starting my PhD, and I was able to buy a new computer that would be able to better support my research. So my research is focused on the premise that cancer is an evolutionary system. So it starts out as a single cell and grows into a population of cells. And just like an animal in the winter has to compete for food to survive, cells in a tumor also compete for space and resources. And This is a process of our ability for cells to adapt to their environment is actually key to understanding why, for example, some patients develop drug resistance. So for example, when you treat with a chemotherapy, you're actually attacking some cells or a population of cells that are sensitive to that, while cells that are resistant have the opportunity to grow out. This is something that I'm really interested in understanding. However, because tumors grow for a very long time, often before they're detected, it's a process that's really hard to directly measure or observe. And so instead, the approach I take uses records left in the genetic code to infer this unseen history. Specifically, I am focused on understanding this process in prostate cancer. While Trevor and I are in different fields, we use a lot of the same tools to understand how genetic changes occur in a population. So for example, the same tools that could be used to track SARS-CoV-2 from the United States to Europe can be used to track the migration of a cell from one location of the tumor to another. As a member of the Bedford group, I have been able to see that being able to track the SARS-CoV-2 pandemic in real time has had a major impact on public health. And although in oncology, these tools are just starting to emerge to track cancer in real time in the same way, I really believe that this will be something that will be incorporated into personalized medicine in the future. So I'm mentored by Trevor on my project in prostate cancer. He gives me invaluable advice on applying phylogenetic methods to study cancer evolution. In addition, I work closely with Dr. Allison Feeder, who's a faculty at the Genome Sciences department at UW, and we work on understanding spatial temporal dynamics in tumors. In addition, I am mentored by Dr. Mary Claire King, who is both my ARCS donor and also one of the co-directors in the medical scientist program. And she's been able to both advise me directly on my project and also towards success in my PhD in general. And I'm incredibly grateful to Dr. King's advice and mentorship. So in addition to my research, I also really value opportunities to mentor. So I'm a teacher in Girls Who Code, which is a nationwide program to teach introductory coding skills to high school women. And so, for example, over the last 2 semesters, I taught courses in R and Python. And I really value this opportunity because I think that computational skills can be very intimidating, and especially to women that are often underrepresented in computational fields. During the COVID-19 pandemic, I also had the opportunity to give vaccinations at clinics led by King County Public Health and Fred Hutch, and I definitely also valued this opportunity to give back or to have an impact in a direct way. And in the future, I hope to become a physician scientist. So since my research is in cancer genetics, I hope to become an oncologist and also lead research that is directed to impact patient care. And so, for example, I really foresee that evolutionary dynamics will become important in personalized care. And so this is one aspect or avenue that I can see making an impact and that will complement my role as a physician. In the past year, I've been really encouraged by how the scientific community has come together to work tirelessly to address this public health crisis. And although it has revealed some challenges with directly applying scientific findings to medical practice, I've also been encouraged as how rapidly science has worked to tackle and combat SARS-CoV-2. Your commitment to funding research through ARCS endowments and fellowships are integral to training the next generation of scientists that will be able to tackle current and future public health crises. And I'd also like to thank you for your vision and confidence in science.
My name is Avi Kenny, and I'm a third-year biostatistics PhD student here at the UW School of Public Health. I'm originally from New York City, and I did my undergrad at Brown University, where I studied biology, although I switched my major no less than 6 times because there were so many different things I was interested in. So my first exposure to global health came my junior year when I was working for an organization called Gardens for Health International. And Gardens for Health operates in Rwanda and essentially helps to build community gardens for HIV patients in the hopes of decreasing rates of malnutrition, which will in turn make antiretroviral drugs more effective. So when I got back to college that fall, I started looking into different organizations who were doing work in the healthcare space in the developing world. And I was incredibly lucky to land a job after graduating working for an organization called Last Mile Health. And Last Mile Health essentially partners with governments in the developing world to build community health programs where health workers are trained to treat diseases like malaria and pneumonia and screen pregnant women for danger signs and essentially extend the reach of the primary healthcare system. So I've been a math nerd my whole life, and at Last Mile Health, I was able to apply quantitative thinking and data science techniques to real-world problems. So for example, during the 2014 Ebola outbreak, we did an analysis of our mobile health database, and we're trying to figure out whether we could reliably diagnose malaria cases without taking blood from the patient. And so what this essentially allowed us to do was enable our health workers to continue serving their communities without being at risk for Ebola infection. In many ways, this was my dream job, and it was really hard to leave. But the more I learned about statistics and research, the more I realized just how much I didn't know. And so I knew I had to go back to grad school to gain the practical skills I needed for a career in global health. Biostats ended up being a pretty natural choice. And to me, biostatistics is the art and science of figuring out how we can most effectively leverage a given data set to answer scientific questions. And this was precisely the skill set I was trying to gain. So I was totally overjoyed when I was accepted at the University of Washington, not just because it's one of the best biostats departments in the world, but because the faculty here are really focused on practical research that's motivated by real-world public health applications. And to me, purely theoretical research is not particularly interesting. I want my research to have an actual impact on people's lives. And I really believe that the faculty here would push me to sustain this focus, and they really have so far. So my research in particular has to do with the COVID-19 vaccine trial data. Some of you may know that most of the vaccine trial data is being analyzed by a team here in Seattle with some members at the Fred Hutch Research Center and some members at the University of Washington. And I've been working with this team to develop statistical tools to examine the relationship between antibodies in the bloodstream and COVID-19 infection rates. And answering this question could help us design better vaccines in the future and can also potentially help us speed up the vaccine development process. And in general, developing more advanced statistical tools allows researchers like Dr. Bedford to answer the scientific questions they're interested in, but with greater precision, or while making fewer assumptions about the underlying data generating mechanism. So it's been a ton of fun collaborating with this team, and there's already been about a dozen other questions that have spawned off of our initial project. So I'm really looking forward to continuing this work. In part because of ARCS, I've been able to fully focus on my research without having to worry about holding down a second job to support myself. And to me, this has really been one of the greatest benefits of being an ARCS fellow. So in terms of what I want to do after grad school, I'm trying to keep an open mind at this point, but I want to do something that allows me to use data science and statistics to help make the world a better place. And so that could be working for a research institution like the Fred Hutch. It could be working in the nonprofit data science space, or it might be in academia doing global health research. And so for now, I'm just going to focus on my dissertation and see where the world takes me. Outside of grad school, the latest thing I've been into is foraging for wild mushrooms, which is a really fun hobby because it gives you a great excuse to hike off-trail in super remote parts of the Cascades and the Olympic Mountains. And if you get lucky, you'll find some dinner for that night. So the last thing I'd say, again, is just a big thank you to ARCS and to my donors, Doug and Nancy Norberg and Kristen Kennefic, for all the support over the last 3 years. Throughout my career, I'm looking forward to demonstrating the value that ARCS has contributed to my experience here at the UW, and I really hope that all of you at this event, through your giving today, are able to help future fellows have a positive impact on the world.
Hello, my name is Shannon Allen Wiles, and I'm studying immunology and infectious diseases in the College of Veterinary Medicine at Washington State University. I am so pleased to welcome you into the Paul G. Allen School of Global Health and to have this opportunity to talk with you about my research, my passion for mentoring, and the invaluable impact the ARCS community has already had on my academic career. After I graduated high school, I decided to attend Middle Tennessee State University. During my time at MTSU, I completed honors research in medicinal chemistry before successfully defending my honors thesis. This experience helped me realize my love for research and the pursuit to create ideas and identify solutions to problems. However, I discovered my true passion was for microbiology when I took an honors microbiology course as part of my degree requirements. I was fascinated with the concept of non-sentient, microscopic organisms having such a large impact on the world. Hello, global pandemic. This course motivated me to change my major, and I ultimately graduated with a Bachelor's of Science in Biochemistry. Before applying to graduate school, I worked for 2 years as a laboratory technician in quality assurance and general lab management. As I continued to enjoy microbiology and molecular biology research, I applied to graduate schools in the biological sciences across the United States. I ultimately decided to attend WSU, where my husband had been offered a master's position. My top 3 offers were WSU, Virginia Tech, and Clemson. When I received my offer letter from WSU and realized I would be an ARCS fellow, I was absolutely ecstatic. Had I not already decided on WSU, this definitely would have been the deciding factor. At the time, I had no idea just how valuable the ARCS community network would be in providing me with an audience of industries, academic colleagues, and other ARCS fellows to call on as resources. I am so grateful for my donor, the Avon Foundation, for supporting my scientific career. In my research, I study the mechanisms which bacteria employ in order to evade immune detection and cause disease, with the ultimate goal of designing treatments which can serve as alternatives to antibiotics. As the threat of antibiotic resistance increases, so does the demand for alternative therapeutics. The World Health Organization estimates that by 2050, the annual death rate due to antimicrobial resistant infections will increase to 10 million deaths per year. Most antibiotics either directly kill or inhibit the growth of bacteria at one specific target point. For example, doxycycline, a common antibiotic used to treat a wide variety of bacterial infections from pneumonia to STIs, works by binding to a single important piece of machinery involved in bacterial protein synthesis. This inhibits the bacteria's ability to produce the proteins that they need, and the bacteria eventually die. Because the antibiotic targets the bacterium at one specific point, this means the bacterium only needs to acquire one random genetic mutation to evolve resistance and render the antibiotic ineffective. My research aims to design drugs which can clear bacteria in a host-targeted manner by enhancing host immune responses, which can then clear bacteria at multiple points, significantly reducing the probability that the bacteria will evolve resistance. My lab has identified 2 chemical compounds, which my data suggests clear bacteria such as Salmonella, Listeria, and Francisella in this host-targeted manner. This means that the drugs clear bacteria during intracellular infection, but have no direct effect on the bacteria when grown in bacterial culture. Both of these drugs are good candidates for alternative therapeutics with low cytotoxicity, no carcinogenicity, and one already showing promise in vivo. In determining the mechanisms of these drugs, we can learn a lot about the mechanisms which bacteria use to manipulate their hosts and cause disease, as Dr. Bedford described in his keynote remarks. And using this knowledge, we can design additional alternative therapeutics to target difficult to treat, multi-drug resistant bacterial infections. I have been fortunate enough to have several excellent mentors. My PI allows me to explore the research directions in which I am most interested, and I've also had the opportunity to engage with several other PIs as my project is quite interdisciplinary. Last semester, I was also fortunate enough to be a teaching assistant for a microbiology and molecular biology teaching lab. And through this experience, not only was I able to share my love of biology with future scientists, I also worked closely with students to design and perform experiments of their own. It was extremely rewarding seeing undergraduate students so excited to do research. My career options are fairly open at this point. I have been awarded the NIH Protein Biotechnology Training Grant, through which I have the opportunity to perform an 8 to 12-week industrial internship with Xylo Therapeutics. And I want to sincerely thank the ARCS community who played an integral role in my securing this incredible internship by using their contacts with industry partners to secure me an interview with Xylo Therapeutics. Some of my colleagues have spent a year trying to secure an internship, and with ARCS help, it only took me about a week. My experience as an NIH protein biotechnology trainee will allow me the opportunity to evaluate industry as a potential career option. I am also interested in a government research position or a position as an academic senior scientist. As a scientist, I am hopeful that as science and science policy advance, we will discover and create new methods of addressing global issues such as multi-drug resistance, climate change, and access to clean water. However, what I'm most hopeful and passionate about is the future generations. I believe that educating and inspiring children from a young age to be excited about science as I was is the key to a healthier, more sustainable future. The networking, mentoring, confidence building, and financial support that you provide through the ARCS community will support these young adults in furthering science and changing the direction of our planet. I want to thank each of you for my own experience as an ARCS Fellow and for the future generations behind me who might be as fortunate as I, thanks to your vision, generosity, and role in supporting the future of science through your contributions as well as your engagement in this community.
I'm Elizabeth Borgeson, and I'm a fourth-year graduate student in the Immunology and Infectious Diseases program at WSU. I am in Dr. Jean Selly's lab in the Paul Allen School for Global Health. I was born in Wisconsin, but then I proceeded to move to the Netherlands and then Michigan, and then I finally ended up in Halifax, Nova Scotia, Canada at Dalhousie University, where I did my undergraduate. I started my undergraduate degree in a bachelor's program for vocal music, actually. And so I was trained in vocal pedagogy, history, and theory. I decided that I wanted to go to med school, so I added biology onto my degree with a concentration in cell and microbiology. And so I actually ended up graduating with a double major in biology and music. After doing the biology, I decided that I wanted to pursue my career in science, and so I participated in a research experience for undergraduates at Harvard University in the lab of Dave Waits, where I worked on a project to encapsulate enzymes in small breakable capsules that could be used in detergent and laundry formulations. This experience made me want to pursue a career in research, and so I decided to return to Dalhousie to complete a thesis project. I worked at the Canadian Centre for Vaccinology in the lab of Dr. Scott Halperin and Song Lee, and I worked on the bacteria Bordetella pertussis, the causative agent of whooping cough. Following completion of my thesis, I returned to Detroit, Michigan, where my parents were living at the time, and I worked at the chemical company BASF, where I once again worked on encapsulating enzymes to be used in detergent formulations. It was at this time that I was accepted to WSU. I was really excited about being accepted to WSU, and the ARCS Fellowship definitely sealed the deal for me because I had two competing offers. I was very inspired by this woman-led organization and also by the organization's enthusiastic commitment to graduate research. Both of these aspects made me feel very excited to be able to represent this program for the duration of my career. The ARCS Fellowship allowed me to focus solely on my research, and most recently allowed me to purchase software and electronics to help me more effectively work from home. And I'll actually be able to use these products throughout the rest of my career. I thoroughly enjoyed getting to know my donors Lynn and Michael Thompson, as well as Dr. Tom Kawula, who is on my graduate committee and has also mentored a number of ARCS Fellows. I also met my best friend through the program because she was the first person that I got to know at WSU because of ARCS. And I've really enjoyed getting to know the entire ARCS community during our visits to Seattle, and it's really made my experience very positive here in Washington. I study the bacteria Brucella abortus, which is the causative agent of the worldwide zoonotic pathogen Brucellosis. Brucellosis is one of the most commonly encountered pathogens in the world, with an estimated 875,000 cases by the World Health Organization. However, a 2018 report actually states that true global incidences are between 5 and 12 million human cases annually, and the US reports about 100 human cases annually. Brucella infects all mammals, including humans. However, the most economically important and impactful are livestock and canines. In animals, brucellosis causes spontaneous abortion and sterility, which is hugely detrimental to a country's economy. And so a number of countries, including the US, have actually committed a huge amount of financial and scientific efforts to eradicating the disease. Humans can contract Brucella through interaction with animals that are infected or with animal byproducts, making animal handlers and vets and hunters probably the highest at risk. The disease develops into a chronic disease, typically leading to side effects such as endocarditis, osteoarthritis, and in some very bad cases, death. And because of this huge public health threat, the CDC has classified Brucella as a biosafety level 3 pathogen. Currently, there are no effective human vaccines, and so the only course of treatment is antibiotics, but relapses are quite frequent, unfortunately. So Dr. Bedford designed a platform that analyzes viral DNA evolution over time to more accurately predict where and when outbreaks will happen and how to respond to them more effectively. He requires input DNA from known pathogens to generate his output data. My research, although we study bacteria and he studies viruses, it's essentially the same concept. But what we do is we characterize that input DNA. And what I mean by characterize is we understand how the proteins that are made by that DNA interact with their environment. So what I mean by that is how do those proteins allow the bacteria to cause disease? DNA characterized by my lab gives his output data context so that he's able to educate public health officials so that they're able to more effectively respond to outbreaks. I entered graduate school on the NIH Protein Biotechnology Training Grant, which awards students a certificate in biotechnology following the completion of their degree, but it also allows them to do an internship during their time in graduate school. I'm really excited because I'll be able to do my internship at Genentech in San Francisco in the summer of 2022. I've always wanted to work at a company like Genentech because I plan on staying in industry for my future, and I'd also like to stay in the biomedical industry. I was awarded the Ponson Fellowship in 2020 for a research proposal that I submitted. And that fellowship is awarded to graduate students conducting medical research in the state of Washington. I was also awarded the Mr. and Mrs. Delbert Caldwell Graduate Student Scholarship through the College of Veterinary Medicine in 2020. And this is awarded to outstanding students who have been nominated by their advisor. I've been very fortunate to have received 3 grants during my time in graduate school. All of these awards have really helped me focus and accomplish my goals in lab and culminated in a recent paper submission to EMBO Journal. My story wouldn't be complete without the people who enthusiastically encourage my love of science, making me a firm believer in being and having a mentor. I've mentored first-year graduate students who've rotated through my lab, and also recently students transitioning into graduate school during COVID. I think it's an important part of the scientific community to give back, and I really enjoy encouraging and fostering people's enthusiasm in science and creating a welcoming and inclusive environment. I'm very hopeful about the future because of the environmental awareness and dedication that the younger generations have. I'm also very hopeful because of people like the ARCS members who think that science is a worthwhile investment and support the younger generations. I think that people always strive to be better, and I think the energy of the young generations dedicated to generating renewable and sustainable resources, combined with people like the ARCS members who want to support these younger generations, will exceed our expectations in terms of scientific and technological advances and will generate a very sustainable relationship with our planet.
Thanks for joining us, Maya, Abby, Liz, and Shannon, for a brief panel conversation. Glad to see you from a distance.
Hi, Dr. Bedford.
Let's get started with a few questions. Abby, in regards to your work with biomarkers, what is the relationship between the magnitude of antibody responses in immunized people and the length of time since their vaccination?
Yeah, so the research group that I work with just put out a preprint that essentially shows that there's an inverse correlation between antibody levels and rates of COVID-19 infection. And that's not surprising at all because we know that the vaccines work by creating proteins similar to those on the surface of the COVID-19 virus. So that if a viral particle enters our system, these antibodies will flag these and essentially eliminate them from our system and neutralize the disease. And so, again, not surprising, but it's still a very important link to scientifically establish in a very rigorous way. And so my research is actually taking this one step further to try to actually estimate the functional form of this relationship. So we want to know, is this relationship linear in the sense that having a few more antibodies means you're slightly more protected against COVID, or is it more like a step function where once you reach a certain antibody threshold, your protection immediately goes up? And so we're developing all the statistical methods to estimate this function and are hopefully going to actually run this analysis next week on the Johnson & Johnson COVID vaccine trial data. And so we'll know a lot more soon, so stay tuned.
Maya, your mentor, Dr. Mary Claire King, discovered the BRCA1 breast cancer gene, and you're comparatively working to track prostate cancer evolution. What are the inherited genetic effects that play a role in prostate cancer?
Yeah, thanks for your question. Unlike in breast cancer, most prostate cancer cases actually aren't inherited. However, in some cases, it is known to run in families, and so we know that there are genetic factors at play here. And there are some main contributors to familial risk, and one of these is the BRCA1 and 2 breast cancer genes. And although they're mostly known for their role in ovarian and breast cancer, they also cause predisposition to a wide variety of cancers, including prostate cancer, and it can actually lead to a 2 to 4-fold increased risk in prostate cancer in men. In addition, there are these overall cancer predisposition syndromes, such as Lynch syndrome, which is when these patients have a mutation in repairing DNA, and so therefore they're at higher risk of a lot of different types of cancer. For example, colon cancer is the most well known, but prostate cancer is also one of these cancers where these patients get prostate cancer at a higher risk. But yeah, I think these 2 examples are really demonstrating that while we originally discovered these genes in a different cancer, in breast cancer, and Dr. King did a lot of this initial work, we actually were able to then build on this foundation and learn about prostate cancer risk as well as the risk for many other cancer diseases. And so this is really also a guiding principle for my work when I'm studying cancer evolution, is learning about really the basic principles for how cancer grows and spreads, so that not only can we learn about one tumor or one disease type, but many different types of tumors and combat cancer in general.
Shannon, how could advances in immune phenotyping, genetic screening, and biosignatures help guide host-directed therapy?
Thank you for your question, Dr. Bedford. Host-targeted therapies are drugs which enhanced host defense mechanisms to clear bacterial infections rather than targeting the bacteria directly to combat the growing issue of antimicrobial resistance. Drug discovery research projects often begin by screening libraries of compounds for a desired result. For example, the clearance of bacteria or viruses in the host, or the activation of specific immune pathways. As the detection techniques which you mentioned advance, these screens will become more streamlined as it will be easier to detect these desired results. Additionally, another key factor in drug discovery research is the determination of the precise drug mechanism. The detection methods which you just mentioned are pivotal in determining host-targeted drug mechanisms by building models to predict potential drug-binding partners, as well as tracking changes in specific immune pathways in the host in the presence of the drug, such as the production of antibacterial proteins. Lastly, also critical to drug discovery research projects is the determination of the complete impact on the host. As advances in immune phenotyping, genetic screening, and biosignatures advance, it will aid in easier, more accurate determination of adverse impacts in the host, which will guide drug design to limit those adverse effects and improve drug target specificity.
Liz, what does your analysis of genomic sequences of brucellosis teach us about the spread of various strains and or genotypes around the world?
Yeah, thanks for your question. So Brucella is a really interesting bacteria in the way that it's quite ancient and it's evolved with its mammalian host over the past centuries. So we actually have species that we've identified from ancient Egypt and cheese. And as I mentioned, we also have identified numerous species in whales and rodents. And so there's actually a couple different things that we're able to learn by studying the genome of this bacteria about how it's able to evolve over time, because it's evolved in such a special and interesting way. Another thing that we've learned from analyzing the genome of Brucella is that we've been able to identify how Brucella is able to infect its individual hosts. So each species of Brucella is only able to infect a certain host, which is called host tropism. And we've been able to determine that it is through the individual bacterial effectors or bacterial proteins that Brucella translocates or secretes into the host cell that allow it to infect a certain type of mammal. And it's those proteins that I actually study. And so this has given us a really great insight into how specific species can evolve, where new species will perhaps pop up, and the likelihood of these species coming to light.
Thank you, Maya, Shannon, Abby, and Liz, for joining us today to share about your research and scientific work.
Thank you,
Thank you, Dr. Bedford.
To ARCS Foundation's donors, sponsors, and community, thank you for supporting science through your generosity today. It's been my pleasure to join you in this important endeavor. Back to you, Margaret.
We hope that this luncheon leaves you inspired and curious, as our chapter is united by its curiosity and commitment to supporting the fellows you heard here today and all of our fellows on their journey. We are excited to be part of their path forward. The money you give provides not just unrestricted funds, but community, connection, and confidence. The support we provide allows these fellows to determine how best to use the funds to power their journey. Many will use the funds to allow them to move to Washington. Some have used the funds to cover lab expenses, childcare, or even the ability to attend the funeral of a close friend. This type of unrestricted support makes a key difference in recruiting and convincing top talent to stay or come to Washington. In addition, it is the type of funding that alleviates financial barriers that sometimes prevent students from being able to continue their education. I ask that you join us in advancing science in America by giving generously today. On the screen next to me is a breakdown of the different ways to give. We are also excited to share with you that $1,000 gifts will be matched by our board of directors. Online donations may be made by clicking the link located in the YouTube chat. You may also give directly at seattlearcsfoundation.org. Donations by check may also be made payable to ARCS Foundation Seattle chapter and mailed to our address listed on the screen. Thank you for your generosity today, benefiting scientists and research critical for our future. On behalf of all of our fellows and members, thank you for your commitment to advancing science.
Thank you for supporting STEM education and research through your giving today. On behalf of our scholars, our universities, and the whole ARCS community, we thank you.
I weighed it as both a financial benefit, which I cannot thank you enough for, but also an opportunity to build community with other students, but also with people like you who are obviously passionate and believe in science and research, and that means so much to me, and I'm so happy to have found this space.
I never thought someone like me would be able to do something like this. I never thought I'd be on this path.
This will really allow me to dedicate more time and to focus on doing what I love. So yeah, thank you. Thank you a lot.
Once again, I can't thank you enough. I hope we get to meet soon in person when we get through this pandemic.
I'm so excited to start my journey at Wazoo. Thank you again for your support through this ARCS fellowship. It really did make a big difference in me considering what school I was going to end up attending. It's just nice to be able to breathe a little bit easier as a graduate student and focus on your research and, be able to do things that will make me happy and successful with this financial support. So thank you again.
I really want to thank the ARCS community for giving me this opportunity to pursue my dream, to continue to be a lifelong learner, continue my education at a prestigious institution such as the University of Washington.